Good afternoon, everybody. Thanks again for the opportunity to do this, and thank you, Rob, for putting the platform program together. It's an opportunity for us to talk about the breadth of what we're doing and give you folks a sense of the areas of interest for Stuart. I showed this slide earlier today, but I changed the words so you have to pay attention. Stuart, we're developing a first-in-class collagen mimetic peptide. The peptide platform is comprised of nearly 500 proprietary sequences. We've only tested about 25 of them, so we have a lot of work to do in terms of classifying these and understanding the details of these. Some of these are designed for lone therapeutic use. Others are designed to be conjugated to therapeutics, and I'll talk about that in just a little bit. So the way this works, and I highlighted this in my talk earlier today, but at a deeper level of detail, there's a damage cycle that happens in inflammatory diseases. And what happens is that the helical collagen structure is unwound. And when it's unwound, what happens is the cell signaling role that collagen plays in tissue homeostasis starts to reverse. So elevation increases, cell signaling goes in the wrong direction, cell growth and proliferation doesn't happen when it needs to, and there are a host of other factors, including potentially complement activation. So you see MMPs elevated, you see integrins elevated, immune cells are activated, and tissue integrity declines. When we do the CMP repair, the opposite trend starts to happen. The CMPs are selectively binding to those open binding sites on the helical structure. We flip the cell signaling role of the collagen helix back on again, and we get downregulation of MMPs, integrins, inflammation, and then you start to get other positive cell signaling activity going on. It's highly selective, and it reduces pro-inflammatory binding sites. We've tested IL-6 and reactive oxygen species levels, and when we show significant decline in a number of preclinical models. So this will be the only time you're going to hear about quartz crystal microbalance with dissipation monitoring today. This is a surface science technique that's used in engineering to determine a number of things, and we've adapted this to help us prove that the peptides bind very selectively to damaged collagen. And I won't go into the details about what these graphs mean, but the left graph means that it doesn't bind to healthy collagen, and the right graph means that it binds to damaged collagen. So we've got, that selectivity has been an important factor, what we look at, because you worry about fibrosis or buildup of collagen in tissues, and that's not something that we want. This shows that it does not occur. I talked this morning about the safety profile. I won't dwell on this today other than to say that we've conducted a phase 2 clinical trial in dry eye disease, and we've done tox studies prior to that, additional tox studies for the phase 3 dry eye trial that starts this next month. And we've seen no effects of the peptide, no negative effects of the peptides in tissues whatsoever, and the adverse events in the dry eye trial were minimal and comparable to vehicle. So our target is helical collagen. It's a previously undruggable target, as they say, and it's ubiquitous in the body. There's 30% of the ECM in the body is collagen. It plays an active role in tissues in the eye from the anterior segment to the posterior segment, and including refractive indications. And the partial list of the targets is shown on the right, everything from Bowman's layer all the way down to the vascular endothelial linings in the choroid. The literature describes the relationship between collagen and the surrounding cellular tissues as dynamic reciprocity. Cells interact with collagen, cells produce collagen, collagen helps control cell behavior, and as we age, our ability to produce collagen starts to decline, and that means that when at a certain point you reach a tip over, and disease indications can become chronic because the body can't produce new collagen to help control the cellular microenvironment. We've established the impact of the CMP on these microenvironments in a number of different areas. Therapeutic benefit in multiple cell types, neuronal cells in neuroprotection, epithelial cells in the cornea, endothelial cells in the vasculature, and we've seen recovery in each of those cell types with the application of CMP. I talked this morning about myopia, restoration of tissue integrity, and the ability to rapidly repair structural collagen. In our first test that we conducted in the cornea, we fully excavated the epithelium on mice down into the stromal tissues, put a single drop of the peptide on the mouse eye, and we had complete recovery of the cornea, including organization of the epithelium, in 16 hours. So it's a pretty powerful lever of repair and recovery of tissues. Finally, localization is therapeutic benefit. We did some nerve crush experiments, and we were able to show that wherever the CMP was localizing in the optic nerve was where we were getting recovery of transport of visual information from the retina to the brain at each point in those nerve axons. So the nice thing about this is it helps remove the excess burden of healing and recovery in the body, and it's a nice intervention that gives us a step function improvement in healing. So this is our pipeline. I shared this earlier. We've been able to pull this together since 2019 on about $23 million worth of funding, and we're pretty proud of that. Partnered with Aju Pharm in Asia for dry eye disease. We've got the phase 3 trial starting pretty quickly here. Great partnership with Glaukos and neuroprotection for glaucoma. Confidential indication that we're talking about a partnership relationship right now should be finalized within the month. And then these two new areas of GA and myopia, which we're thrilled about. Specifically in indications in the eye, why does this work? How does this happen? In dry eye disease, our targets are collagen in the cornea and the trigeminal nerve. The status of that program I've already described, and in the fine print here you see the publications that we've done. I'd be happy to share the publications with you after the session here today. We've taken a very aggressive approach and a very thorough approach to publishing the work that we've done in each of the areas of the eye, just to demonstrate something that's fundamentally new to the industry. In glaucoma, our target is collagen in the lamina cribrosa and the optic nerve bundle in the glial lamina. We've partnered with Glaukos there, of course, and in myopia, I talked earlier today about our target being collagen in the posterior sclera, but also potentially in the choroid and Bruch's membrane, because there's some arguments that they play a role in myopic development. I wanted to focus in on the retina for just a minute as a specific area, because this is an area that we're quite excited about. We conducted a primate study late last year, and we did a drop formulation. We fluorotagged the CMP, and we applied it to a primate to see what was going on, where it would localize. And this particular primate had undergone a 6-month elevated IOP program using the microbead model. So the tissues throughout the eye were pretty distressed after that period of time with the microbead model. And where you see the CMP localizing in that center image is in several important and distinct areas. In the synapse layers, definitely see it there. It's very brightly highlighted in the interdigitation zone between the RPEs and the rods and the cones. And then you see it distinctly in the choroid and in Bruch's membrane, and in the vasculature in the choroid as well. And then we can carry this even further back into the sclera. We also have work that we've done in the cornea where we've seen it in animal studies binding to and correcting collagen damage around the trigeminal nerve, and we believe that is why we were able to recover tear function in the eyes in our phase 2 clinical trial, because nerve damage in the cornea led to nerve damage in the V1 trigeminal nerve loop for the lacrimal functional unit. Finally, one more area of flexibility is the ability to bind therapeutics to this, use it as a drug delivery platform. We've got very flexible binding capability with the peptide. We can use any number of different bridges or extensions or design approaches to binding and delivery of a therapeutic. And we think this is a particularly effective approach in certain areas that gives us a synergistic benefit of the API itself, as well as the therapeutic benefit of the CMP on the damaged collagen in the region that we're targeting. We've got clinical efficacy in our phase 2 trial. We've got a dual MOA that we're excited about. The target is critical to multiple tissues in the eye that are implicated in the various diseases that we're all concerned about. It's highly selective. We've seen no off-target effects, and it's got a strong safety profile. And there's a huge opportunity for us in this world. We just need to be selective about what we go after. One of the largest opportunities in ophthalmology, we believe, and we're thrilled to do this, and we'll love to come back and see you guys again next year and let you know how it's going. Thank you very much.